to the departmental seminar. This is the third to the last one. It's coming down to the end. Keep the one 2 weeks from today in mind in particular, because that's when we will have our departmental celebration. So that one's a fun time. We'll have a little more technical seminar next week. Today I'd like to welcome Nancy Plackage from the Department of Microbiology and Immunology. So he'll be talking about another application area specific to his research, but incorporating machine learning, which again fits within our role in data science. So with that, thanks.
Thank you, John, and thank you for the invitation. I'm not a computer scientist. I don't have any degree in computer science or anything like that. But as you'll see from my next slide, I've been here, by the way, about 10, 11 years. So I came kind of in early 2000s. Been a while. And my department used to be microbiology when I came here first, but now after the merger, it's immunology as well. So I'll do a shameless plug right off the bat. So I teach a course called Advanced Bioinformatics. It's going on as we speak, actually, and it's on even years. But I'm trying to convince my boss, my department head, to move it so that it's every year. And basically, for you guys, this would be a chance to learn some biology. I used to actually have a couple of computer science students take it as a course over the years, but it's mostly biologists that take it. And there are different things here, some of which we'll touch upon in this talk. So, being a biologist, or more accurately, molecular biologist, I'm interested in how to apply these techniques to biology. And actually, this is how the course is geared towards teaching biologists how to use computers. So, from my perspective, this is kind of a secondary area of research. It's not. I mean, I have a real experimental lab, and we do all kinds of different things. But that said, this is a really big interest of mine. In general, understanding relationships between proteins and protein families, and it's still an unresolved area in biology to a good degree, as you will see as we go through the talk. So I'll tell you first about sequence comparisons and why we're doing those, and especially I'll tell you about some of the recent developments, specifically profile methods for doing these sequence comparisons. Then we'll see how we can actually detect protein homology relationship or lack thereof using just protein sequences. Towards the end, I'll just touch upon doing the same, but now using protein structures. And then we'll see how everything that's kind of bundled into this talk, how it can be used in the end to improve functional annotations of proteins. So the whole thing, there's a misconception where bioinformatics started. We kind of can pinpoint when maybe the term became abundant. And let's say that it was at some point in mid to late 90s. And that coincides really well with the sequencing of the first full bacterial genome, which was Haemophilus influenzae. Now when we look at that genome, this genome is less than 2 million bases, has less than 2,000 genes. If you look, this is literally, I put this on purpose, a list of all the authors when that paper was published, and you can see there's 20-something people sequencing a tiny, what by today's standards is tiny bacterial genome. Literally, a student or technician can do this now in 2-3 hours of one day and probably annotate the genome in another couple of hours. But back then in 1995, this was a big deal. And you can see that the pace was very, very rapid. So we started in 95 by a very small genome that was only about 1.5 million bases. A couple of years after that, we were already order of magnitude above that. So we were already doing yeast genome, and you can see now this is almost 15 million bases. Again, a couple of years later, we move another order of magnitude up. Now we're at hundreds of millions. So this was now a worm that was sequenced. And then basically by 99, there was lots of genome blocks. 30. I mean, today we have more than, today we have literally thousands of genome. But you can see how in these 4 or 5 years, the sequencing picked up rapidly. In other words, it's not a problem today anymore to make a lot of to sequence lots of DNA. That's absolutely trivial today. And as Greg Pesco remarked, uh, and about that time when I said that sequencing has picked up, genome sequences now accumulate so fast that even an average lab can produce in a day or in a week more data than Shakespeare produced during his lifetime, although rightfully so, as he remarks, Shakespeare is probably a better read than just reading random strings of A's, C's, G's, and T's. But nevertheless, today now that's our. So where is actually then the bottleneck? So we can make sequence however we want to. The problem is annotating those sequences. That's where the bottleneck is. So we get our genomic sequences, we convert them into proteins, so we now get protein sequences. Now we need to annotate them. The good news is we can do the majority of those very, very easily. So the majority we take care of. Unfortunately, 30 to 40% of all of those sequences still do not have any functional or any meaningful, I should say, functional annotation. What that means is basically we know the protein sequence, we know that it's shared by many species, we don't know what actually that protein is doing. We don't even have a clue. That's becoming a bigger problem now than actually getting the sequence to work. The reason for that is that in nature, protein sequence is not very well conserved. When we're talking about proteins, proteins can mutate quite a bit and still retain their general structure and function. So structure is much more conserved than sequence. In other words, you can have two protein sequences. If you pick ATP synthase from human and from yeast, they're going to be considerably different. Yet they'll make a protein that looks very, very similar and performs the same general function. So because of that difficulty, that's why lots of these proteins we actually cannot uh, annotate. So those that we can, how we're doing that, we're doing it by comparing sequences. Comparing sequences really predates bioinformatics by a lot. I mean, even back to if we go back to 1960s and especially 70s, there were already methods for aligning and scoring strings of letters. That's literally what sequence searching is. So the idea is to identify all the homologs. If we have a protein A and we do a sequence search, the general thing that we're trying to do is identify everything that's related to it, true positives or true homologs. If we can do that, and we know the function of protein A, we can it's a guild by association. We can basically infer the function of protein B simply because it's similar enough to protein A. That's like if you see someone athletic looking and they tell you that they're triathletes and somebody stands up to them next to them that looks the same, you infer that they're triathletes. No, I'm making a joke here. But as a general rule, it has to be some kind of measurable, tangible similarity, not just superficial similarity, for us to be convinced that this is the case. In doing this, we also want to limit the number of non-homologs or false positives. Sometimes that would be the example that I just gave. Just because two people look similar doesn't mean that they're both athletes or that they're both good cooks or anything like that. They simply look similar. And then lastly, we want methods that can search the database efficiently. Uh, when I started looking at protein sequences now, which is now my age, let's say 15, 20 years ago, the way to search a sequence database was to send an email to a server, and then the server would look at that email and extract sequence from it and do the search and then send you an email back. Um, that's how you would search for a database. And that could take anywhere from 3-4 hours to a day. And of course, today it's a matter of seconds. So again, by doing these sequence comparisons, we get some kind of a score. This score is usually, there's some kind of a scoring function that converts that score into something that we can interpret in terms of statistics. That's usually an e-value. And you'll see in a second how we do that scoring and actually how we, this is just kind of a general intro just so we have a setup for what happens later. So here's an example of a simple blast search, and here's an alignment of our query and whatever it found in the database. And this is only a partial alignment, by the way, it's not a whole thing. And you can see here that it has a score 107 bits, that it has p-value of e to the minus 23, which is highly statistically significant. Any e-value basically lower than 0.01 is generally considered statistically significant. So we can either make a global or local alignment. BLAST actually generates local alignments. As a general rule, local alignments are better for scoring. They behave better than global alignments, but it doesn't matter. Those are just details. Just briefly, if you're wondering how this is made, again, this has been worked out in the 1960s and 70s. It's a simple alignment of strings by dynamic programming. So if you have a string number 1 or X, string Y, and you're trying to align them, and let's say you're a simple scoring system where you'll give 5 points for a match, you'll give -2 for a mismatch, and you'll give a -6 points for a gap. A simple way to do that is to put one of these words up top, the other word here on the side. Start from this corner and start filling in this matrix by numbers. If it's a match, you add 5 to the score. If it's a mismatch, you subtract 2. If it's a gap, you subtract 6. So going from here to here, M and M, that's a match, that's 5. Now if you want to put an insert in here, that would be -6, so -5 becomes -1, and so forth. Basically, computers are good at these kinds of things, filling in this table instantly. And when they do that, the only thing you have to do is find the maximum score in this, which would be this, 29, and then find the shortest path from this end to this end, which in this case happens to be almost a perfect diagonal because we're comparing two similar words, but as a general rule, it doesn't have to be a perfect diagonal. It can be some going up and left and right, depending on where the insertions and mismatches are. So basically, this was a very simple and unrealistic scoring scheme. And basically, a more realistic scoring scheme looks like this. I don't know how many of you are using this on a regular basis. This is a Blossom Substitution Matrix. Some of you are using it, actually. So this is a matrix that tells you for any kind of alignment what kind of score will be given. So if E is aligned against A, that would be -1. If E is aligned against H, that would be 0. 0 means that's a neutral substitution. If it has a negative value, it means it's an unlikely substitution. In other words, in proteins, E is unlikely to be replaced by A, but E is likely to be replaced by D, because you can see E and D, those are both negatively charged amino acids. They can substitute for one another pretty good. So that's it. We have a scoring scheme. We have a dynamic programming algorithm. We now go and score sequences. We can now align any two sequences, right? Well, maybe. So I'll take my word for it that these two sequences are related to one another. The one on top, this is literally an exercise that I did when I came here and started collaborating with Mark Young. Some of you I'm sure know it. So this is one of his viral sequences that basically, if you search against the database, you would not find anything meaningful. Now, what's underneath this is a is a bacterial transcription factor, a protein of a very defined and known function. So I put them like this, align them, and every time you see the vertical line, that means that you have an identity. Now I'll save you the counting. There's 11 identities here in a protein that's 43 amino acids long. So we're talking about 25% identity, 26 something percent identity. Okay? Do you think this gets scored as related? So when we actually use a scoring by BLAST or by some other alignment methods, then this will be scored as related. So here it is. If you ask BLAST to align these two sequences, it'll give you an e-value of 14,380. In other words, it's a decidedly no relationship. That's what this e-value is telling us. Definitely no relationship between these two proteins. Now you can change, that was Blossom substitution matrix, you can change it to 45, and now you'll get an e-value of 34,000. Again, very, very isolated. And you can keep doing this. You can change it to Blossom 80, 2,000. You can change it to PAM 70, almost 7,000. In other words, if you use this literally, to search, to compare these two sequences, and if you literally believe what the e-value is telling you, you will conclude that these two proteins are unrelated. I'm just giving you one example. This is just one of many examples where actually these general substitution matrices fail. So why did they fail? That's the question. So you're now taking my word for it that these are related. This has since been proven. They're definitely related. But why did the matrices fail? Well, first of all, these substitution matrices are meant to be general, meaning they're meant to work on the majority of the protein pairs out there in the database. That's the meaning of a general substitution matrix. They don't work 100%. So if you're comparing two protein sequences that have diverged over a long time, that are separated by long evolutionary times, they can diverge so much that basically they cannot be scored properly anymore. That these general matrices will score them as statistically insignificant. And as I say here, these substitution matrices also fail because they work better on some protein families rather than the others. So the question now is, can we build a protein-specific and position-specific scoring scheme? So let's say we have a protein, and when we search against the database, we don't find anything meaningful. But if we had a meaningful scoring matrix that's specific to our protein, then hopefully we would be able to find other matches. And that brings us to sequence profile comparison. So what I showed you earlier on the BLAST example was comparing a single sequence against a single sequence. So now those are sequence-to-sequence comparisons. In this case, we have a sequence-to-profile comparison. I'll show you in a second what it is. So we start with our protein. We do the first search using the regular matrix, and these are the three proteins that we find in addition to this one. Okay? So this is real life example. This is exactly how it happened 10 years ago or whenever I did this exercise the first time. Basically, you can now align these four proteins, okay? And you can build a substitution matrix based on this alignment. Because at each of these positions, you can see that at this position, V is invariant. But then another V, let's see, is not invariant at this position. In other words, this position should be scored differently than this one. Because in one of them, a given amino acid is very invariant and the other one it isn't. A good example is this double K. So this KK from our original sequence, you can see how it's substituted one way at that position, and then it's substituted differently at the other position. Which is to say that they should not be treated as the same residue. They should be treated as different residues. So we take this alignment, we build the protein-specific matrix or profile, Now we search. So here's how that looks in practice. How many of you are actually using SciBlast? Probably. So here's exactly, we do the search with single sequence, we find these four, and then we build the alignment based on these four sequences and click on this, run iteration 2, and before I show you the result of the iteration, pay attention to the sequence that's still considered significant versus the sequence that's just below the threshold of 0.01 for the value. If you look at them side by side, so here's the alignment of the last significant sequence, here's the alignment of the one that's just below the threshold. There really is no significant difference. I mean, is this alignment better than this alignment? Apparently, scoring-wise, it is better. But really, is this alignment more convincing than this alignment? It isn't. I mean, they are about the same length. There are no gaps in either one of them. Yet again, this one scores as statistically significant. This one scores as statistically insignificant. So now if you build a new scoring scheme from the original alignment, what will happen in the second iteration, this sequence will now actually become statistically significant. It'll, in the second search iteration, it'll get a significant score. You get the idea. If you keep repeating this, if you now build the alignment from these 6 sequences as opposed to our initial 4, you'll get a new scoring matrix, and you can keep repeating this. SciBlast does this on the fly, and that's how it improves the detection. And then in the 4th iteration or the 3rd, it'll detect another 3 or 4 proteins, and you can now keep going on and on and on for as long as you want. So in the end, it'll be actually a long list. And if you read some of these annotations here, let's see, putative transcriptional regulator. Actually, what I told you earlier, that those two are related, they definitely can be proven to be related. The last and greatest variant of this is what's known as a profile scoring. So just to again make a comparison, in the original sequence search, we were comparing a single sequence versus a single sequence. In the profile, which I just showed you, in the profile to sequence comparison, we're comparing an alignment of sequences to a single sequence. And then the latest and greatest incarnation of that is to take an alignment and compare it to another alignment. Literally to score multiple sequences versus another set of multiple sequences, which looks like this. If this is our query about this black line, and these are logo representations, so you see what residues are there and what their frequencies are, and this is underneath this line is now a logo representation of our match that we're aligning. Now you're literally comparing this whole column to this whole column. And throughout the alignment, you're doing the same thing, rather than comparing a single residue against another single residue. And needless to say, this improves the scoring quite a bit. It improves the detection of homologs. So this is a slower, of all the implementation of sequence searches, this is the slowest, obviously. There's most to do, but it detects lots of distant relationships. A program that does that, by the way, it's called HHSearch, and I'll use it throughout this talk. So it would seem like, yeah, now we have great methods, everything's looking great. We just improved the detection of homology. Yet, when we now plot this and see how many of these true relationships are actually being detected, we'll see that quite a few of them are missed. So this is probably an unusual plot for you, so let me just explain it. So there are many different ways to plot the effectiveness of any given search method. But what I'm doing here is I'm plotting the false discovery rate, which is basically how many false positives per 100 do we have. So if a method makes 100 alignments, how many of those, and scores them, how many of those will be false positives? Okay, so So that's what this is. And on the y-axis we have coverage. So for a database that I'll explain later, there is obviously 100% coverage would mean that every single positive relationship that exists between the protein, if we can find all of them, then the coverage would be 1. So you can see that using these methods, how the coverage monotonically increases as you increase the false positive rate. So this, by the way, explains you why we are using p-value of 0.01. Because you can see if you look at the triangle, at that p-value, we have about 1% false positive rate. In other words, at that particular p-value, 1 in 100 alignments will be scored wrong. 1% is actually more than what most biologists would like to accept. I know there are lots of methods where people go even up to 5%, and if you went to 5% here, that would give you a coverage of almost 80.85 or 85%. The problem is, in biology, too much depends on correct annotation. In other words, if I misannotate 5 out of 100 proteins, there are going to be labs out there that believe my annotations, don't go do the experiments. Those experiments will fail because 5% of my annotations are incorrect. In medical professions, there are people who use these annotations. They'll be wrong as well. 5% is just way too high. So like I said, there is a reason why p-value of 0.01 is chosen as a cutoff, even though there's only 75% coverage. Basically, only 3/4 of all the matches are detected. But at least at that point, we have just under 1% false positives. So our goal is then, whatever we're doing is to keep increasing the coverage, which is not just a different word for the sensitivity, while decreasing the false positive rate, and that basically means having a high specificity. So we want curves that will be above this one and not go as far to the right. If we can get those, that means we're actually having a better scoring system. So just to reiterate, does it always mean if we align two sequences and we get statistically insignificant p-value for them, does that mean that they're not related? Most of the time it does, but it doesn't always mean that they are actually unrelated. As you'll see later at the end of the talk, unfortunately the same is true for structures. We can align two structures and again get some kind of statistically insignificant value for that alignment, and again it may turn out that our scoring is just not proper. Okay, so here's now kind of, again, hands-on explanation why sometimes these methods don't work. So here's the protein. It doesn't matter. This XSH it's responsible for the metabolism of histidine, amino acid histidine. It doesn't matter. We're searching using this protein against a database that actually contains this protein. So if you pay attention to the e-value column, this is now all the e-values. You'll see that there is literally one match that has statistically significant e-value. In other words, in the whole database, this protein only matches itself. It doesn't match anything. All of the other matches have statistically insignificant e-values. Okay? Now, does that mean that this protein has absolutely no relatives in this database? And the answer is no, it does not. So I'll show you the alignment for the hit number 2. So here's the alignment for the hit number 2. And again, if you look at its e-value, it's 0.8. And an expert, a person who's doing the annotation, can actually look at this alignment and instantly tell you that these two sequences are related. Why would that be? Anyone? Come on, give it a try. Here, I'll give you a hint. Do you get a long alignment if the sequences are unrelated? No. The longer the alignment is, the less likely it is that sequences are actually unrelated. You'll get 5, 10, 15, 20 residues aligned just by chance between two unrelated proteins. But if you have here you have 81. If you have 81, that alone is very, very unlikely to happen for proteins that are unrelated. The second thing that you do, again, by visualizing is compare secondary structures of, of our query to the hit. So H means helix, E means extended, that would be actually a beta strand. So if you do that, you can see that this helix matches that helix, that this strand matches that strand, that strand matches that strand. You can see the pattern that in terms of secondary structure, these two sequences match extremely well, which again, doesn't happen randomly. You don't get two sequences that match over a long period and by and have their secondary structural elements matched by chance. It just doesn't happen. And again, strand and strand and helix align. So someone who would look at this alignment would tell you instantly these are most likely to be related. And now in particular, if you know something about this group of proteins, that this B here is actually a catalytic residue, and then you scroll down and look, this one has the same catalytic residue. There's a K, there's an E here that's also catalytic. If you look at that E and scroll down, this protein has E as well. So everything about this alignment screams that they are related. The catalytic residues are preserved, the length is preserved, secondary structural elements are preserved, yet we just cannot prove it. E value says that they're not. Okay? So just as an exercise, this is now number 12. So I'm picking one off the bottom almost that has E value of 28. Again, completely unrelated, one would think. But if you do the same exercise, compare secondary structures, length of the alignment is now over 100, it's pretty obvious that they are actually related. So what I'm doing here is I'm using my own expertise to figure out the two sequences are related even though E value says that they're not. Doesn't it sound like a setup for some kind of machine learning approach? Where basically the idea is to train a computer to use similar kind of logic to figure out that two sequences are related even though their p-values said that they're not. That's basically what we're talking about. So how can we convince ourselves? How can we convince anyone out there? Let's say I'm hand waving to John and say these two sequences are related. I have a gut feeling. My gut feeling amounts to nothing, really. The point is it has to be somehow proven. So one way of doing it, and as an experimentalist, I would definitely try it, to do the experiment and show maybe mutate those catalytic residues, but that takes time. That takes anywhere from a couple of months to maybe even a year. The second thing is, again, use human expertise. So that's the exercise that I just did. But the third one is, let's train the computer to do this part for us, to basically extract these patterns that indicate the two sequences are related, even though their e-value says that they're not. So like for any machine learning approach, we have to, this is a supervised machine learning, we have to have a labeled data set where we know that protein A and B are related and protein A and C are not related. And preferably, we need to have a diverse data set that has lots of sequences that are obviously related and then lots that are not so obviously related. So this is a database of protein structures. It's called SCOP. It says Structural Classification of Proteins. And it's a manually curated database. So now, for each protein pair in the database, it's manually curated that they're actually related or not related. And so there's about 39,000 protein structures. There's more than 100,000 individual domains. So that's a lot of data points. And then in this classification, there are families, superfamilies, and folds. Let's just say, actually, I have it on the next slide, that we're interested in superfamilies. So at the level of family, two proteins are definitely related, evolutionarily speaking. At the level of superfamily, they're also definitely related, but their sequence identity is usually poor. It's usually below 30%, which means that it's difficult to prove the relationship at that level. And finally, at the level of fold, proteins have the same general shape, but they're not necessarily related. Most of the time, they're not related at all. So, knowing all of this, I can build. Uh, Start with my ground truth, which is now abstract sequences from this database. I know label them for any pair of sequences. They're either 1 if they're related, or they're 0 if they're unrelated. Okay. So I started this by using support vector machines, but as you'll see later, I tried a bunch of other things and all of that work to one degree or another. So we'll use these profile, meaning we'll score alignment against alignment, we'll get p-values, and then we'll see after we train this classifier what actually happens. So just a brief reminder, support vector machines are maximum margin classifiers. So this is an easy example. This is basically a bunch of just in data points that are easy to separate linearly in 2 dimensions. But the goal of it is to find the maximum separation. So any of these lines that are drawn through this data set would actually separate them. But the red one is the one that gives the maximum margin uh, between the plane itself, the separating plane, and the next data point. So I'm going to skip this. So what do we then use for training? So in order to train the classifier, we have to extract feature. So we use the output of this profile comparison, and we'll use obviously e-values. Actually, rather than using e-values, I'm using p-values here because p-values are independent of the database length, while e-values are not. But it's the same meaning. E-values are derived from p-values. It's just, this one's just more convenient. The second thing is this program already gives you what it calls a probability, although it's not in the strict sense a probability. It gives you a probability that this query and this hit are actually related, and you can see that's another feature you can extract. Third feature you can extract is this SS, which is a secondary structure match. Remember when I was showing you how helices and strands match? Well, this program actually captures that in a single score, just a single number. So that can be used for classification too. We can see then the next thing we can do is for this query, we know that there's 268 residues, and then in the template, how many of these are matched in the template? And basically, we can derive a fraction. In other words, is 5% of my query aligned, or is it 95? If 95% of my query is aligned with the target, then that's more likely to be true than if only 30% of it is aligned. In other words, from all of these numbers that are listed in this output, lots of features can be pulled out and then used for classification. So this just basically tells you build profiles for all of these proteins. There were about 15,000 of them. Compare all of them, so do the HH search, compare profiles to profiles, get about 15,000 of these outputs, like the one that I just showed you, extract these features, and then after we do all of that, we end up at about 2.5 million data points, where about 1.5 million are true positives, and about another million are true negatives. So it's not exactly one-to-one ratio between positives and negatives, but it's pretty close. Just if you're wondering, whenever you're deciding to classify something, I highly recommend that you do some kind of dimensionality reduction just to see whether your data set is actually classifiable to begin with. So what's shown here is principal component analysis of the whole data set. And you can see it's plotted just in the top 2 principal components. You can see that the first principal component explains 75% of the variance in data, which means a single, that would be p-value. P-value alone is enough to classify data very, very accurately. Add to that the second principal component, which explains almost 12% of the variance. Between the first two principal components, we're basically able to explain almost 90% of our data. In other words, that means this is a data set that's highly classifiable. It definitely is something that can be classified and classified well. So this shows you basically related proteins versus unrelated proteins. And it seems like there's an overlap, there's a funky kind of overlap and funky shape, but trust me, I mean, that's what the plot's saying. This is something that's easily classifiable. So if you do a simple classification by e-values, or in this case p-values, this is the accuracy that you get. You basically get about 95% accuracy. That's not bad. You're literally using a single column, you're using just p-value. If you actually now build a classifier using the p-value and using that probability column, that improves the classification a smidge, not even a whole percent, just a fraction of a percent. If you now start adding secondary structure scores, now it jumps by about 1% in accuracy. So you can see most of the classification power is already in e-value, but nevertheless, there are features if you keep adding feature after feature that actually improve the classification. So you can add those fractions of a line query and the hit. That improves. That's basically what's shown here, number 3 and number 4. And then you can use another method and combine. So this scoop is just another sequence comparison method. So you can combine scores from one method and another, and that'll give you even better classification. You see these numbers. These are kind of silly numbers. I mean, most of the time you don't get 99 plus classification accuracy. But again, keep in mind, e-value alone is very, very deterministic. So all of these other features are just adding little by little, pushing the score towards 100. So if you do the ROC curve or curves for all of these classifiers, you can see actually that they're all excellent. A perfect curve would basically go against the y-axis. So if you do the blow up just of the upper right corner, you can see there are differences between them, but basically each of these classifiers is very, very good. So now that we have that, can we use that somehow to annotate proteins that are otherwise not annotated? So if you now take lots of these proteins and build this graph, this is just a fraction of a protein database, just a representative to make my point. So you can see in the legend here, the blue means that this classifier gives you the probability between 0.7 and 0.8. Green is 0.8 to 0.9, and then a red one is the classifier gives it a probability larger than 0.9 and they're related. So if you build this connected graph, okay, what you can do is, again, by association, annotate all of these proteins. So all of these proteins that I've put out on the edge, they're all proteins of unknown function. All of these proteins that are on the inside are restriction nucleases or some variant, some kind of a nuclease. And when you see how well connected they are, you can literally infer, again, built by association, that each of these proteins that are drawn on the outside are actually some kind of nuclease as well. Because each of them has multiple confident connections from proteins that are of known function. The idea again here is that if you have enough of these connections between known proteins of known function, to proteins of unknown function, you can actually now start annotating all of these proteins. So it shows you here everything that's in red. I don't know if you can read from there, I'll read for you. Most of them are uncharacterized protein, or predicted nucleotide binding protein, or protein of unknown function. You get the idea. Most of these red ones are actually proteins of unknown function. Yet in a connected graph like that, especially when you see how many connections, I mean, pick this one. It has 1, 2, 3, 4 confident connections to proteins of known function. You can basically annotate every single one of them. So that was the first attempt at this that I did 6, 7 years ago now. Then the idea is then you get greedy. It's like, okay, lots of these. Are there any questions? I may be going to ask. Real good? Oh, so can we build a better classifier then? Yes. The answer is yes. We can try other machine learning methods. So I tried neural networks. They don't do a better classifier. Then I tried ensemble methods. So we can do bagging, it works better. We can do boosting, you can make an ensemble of the single trees, basically boost that, and that works great. So what happens, if you remember, this is our plot, how it looks like. What happens is that after we use this classifier, this is the curve that we get. In other words, we get a way better coverage, pushing now 90% as opposed to what was here. And you can see that the false discovery rate is now extremely low. This is what you want. You want a classifier that has high sensitivity and high specificity as well, meaning it errs very, very little as a general rule. If you again plot rock curve, this looks like an empty graph. The curve is actually right here. I mean, it's just about perfect. You can see area under curve is just about 1. So the problem here is that, again, we're still under 90%. Why are we under 90%? That's because we cannot get enough true positives in the output to classify them. Basically, it's classifying everything that's in the output correctly, but the output simply contains less than 90% of all the possible matches, again, for scoring reasons. So how can we get more true positives? One of them would be to increase the e-value when we're doing a search. Now that brings in very few extra positives and brings literally millions of negatives. So it makes it difficult to classify. But it can be done. This is an imbalanced data set, but by fiddling a little bit with it, we can get a second classifier that's now pushing 90% coverage. So we're moving little by little. Now, if you're, again, that's a great classifier. If you look at the area under curve, it's just about perfect. So I call this, again, glass half full. I should be happy that I have a perfect classifier, but actually I'm unhappy that my coverage is still only 0.9. What do I do? How do I squeeze out these 10%? So the last thing that we did here was basically try to figure out an indirect way of increasing this accuracy. So this goes like this. It uses the idea of collaborative filtering. So that means, can we indirectly predict if you and I like CDs, and if both of us buy wine, chances are if I like ice cream, you'll like ice cream too. I'm trivializing that, but the idea is if you're ever wondering how Amazon or Google or everything, how they read your mind as you're browsing around and they keep putting things out there for you to buy, they're using that same general idea. No matter how special we are, there is someone out there, many someone, as a matter of fact, who have similar shopping preferences to us. And whatever they shop, if they're similar enough to us, will be recommended to us. That's basically how that works. So does that work for proteins? Basically, here's the idea that I just told you. If Mickey is related to each of these 4 individuals with this score, and we find out that it's mostly related to the bunny, then we can say if both of them like hamburgers and pizza, and we know that Mickey likes ice cream, then we'll say, well, probably bunny will like ice cream too. In other words, if proteins A and B are related to protein C, and protein D is also related to protein C, protein D is likely to be related to A and B as well, even if we can't prove this. So, using the general, using the general idea, we can actually build even a better classifier that has even higher coverage by another 3-4%. Now we're getting well into 90s. The problem is that this classifier has the higher error rate. So in recommender systems like Amazon and such, if they got 9 out of 10 recommendations correct, if they gave you 10 correct recommendations, they'd be crazy happy about that. Here, I'm not so happy with the fact that actually I get better classification, but at the expense of accuracy. So I will wrap up just to tell you that the same thing can be done for protein structures. So even though protein structures, it may seem like, oh, it's a no-brainer, you align to protein structures, you can easily see whether they're related or unrelated. That's actually not true. For protein structures, as well, there's ambiguity for a variety of reasons um, that makes it difficult. So the goal of all of this is to build a database that will annotate all of these uncharacterized protein families. By using these general approaches and clustering proteins together, basically, if I get a cluster in which I have 5 annotated proteins along with another 5 that are annotated, I can literally transfer the annotations from the known ones to the unknown ones. It becomes simply a clustering problem. So this is not important. I'll skip it. And these are the people to thank and the grants, and I'll stop here and stop the questions.
We actually don't have time for any questions, but we'll take one.
So did you guys have a chance to see how accurate your
It is actually, it's very accurate. For most of these things that I've showed here, there is some kind of experimental proof already, meaning the structure was solved or somebody did the experiment and characterized proteins. Honestly, I'm yet to be wrong on anything that I've published. Any of these annotations that are actually published, and quite a few of them are, every single one has been proven correct. There's one that hasn't been proven correct or incorrect. Nobody just did the experiment. But I would say probably dozens of them that are published, they've all. So these are very, very accurate, actually.
Thank you, Mr.
If